Now, DNA fingerprinting we are going to study. Okay, 1 minute. Okay, let's go to learn DNA fingerprinting. It is a laboratory technique used to identify individual by analyzing unique DNA patterns. It have okay, it will performed under the lab. Okay, and to analyze the individual's unique DNA pattern. Okay, here synonyms or some related DNA fingerprinting synonyms is there. That is DNA profiling, DNA typing, identity testing, genotyping, genetic in fingerprinting. It is developed by Sir Alex Jeffreys, okay, in 1984. Principle or what based principle DNA printing will analyze because of the highly variable repetitive DNA sequence. So those highly repetitive DNA sequence called that is mini satellite are variable number of tandem repeat. Those mini satellites are variable number of tandem repeats. These reason are very greatly among the individuals. Those repeat the variable number of tandem repeat that will very great among individual to individual they vary. Okay, exception to remember here, some identical twin have identical DNA fingerprints also. Okay, identical twins have the DNA fingerprinting of the same. Okay, step, what are the steps? Steps are there in DNA fingerprinting process. That is first step, collect sample from the blood, hair, skin, saliva. Okay, whatever you want sample you can collect. After step 2, they extract DNA from the cell, and after that, amplifying DNA using PCR polymerase chain reaction. By using polymerase chain reaction, we amplify the DNA. Amplify means spreading, lengthening. We need to lengthen. Here, step 4 and the sequence of DNA by gel electrophoresis. We analyze and then generate DNA profile. We can generate what is this, what they will do, what are characteristics of these gene. They will finally finally made. I made the comparisons with others. That's it. Key application. Its application is forensic science. It help in forensic science by identifying criminals. If any dispute happen, any crime happen, there's crime scenes, it will help to identify the who are the criminal. Okay, and parenting testing also, father child dispute also, identification of bodies if any in disaster like bomb blast. Okay, in during bomb blast, so many body pieces will spread so many places. Because of those pieces we can get to know that this body belongs to them. It's hearing is horrible, right? Let it be. This is happening every single day in our world. We can't collect. Medical genetics. Wait. Here and then organ transplantation. It will help to organ transplantation, matching donor or recipient. We need to identify, right? And medical genetics, medical genetics also detecting hereditary disease. We can detect hereditary. If any disease of father, that disease will this will be present in the offspring, right? We need to know the offspring disease well came from genetics or not. That also we can detect from DNA fingerprinting. Okay, limitation and challenges here. What is that? We here challenges are there. Sample contamination. We, if possible, sample will contaminate means with other sources. Okay, and human error is handling. The human being who are handling that sample, they may made some error. The improper interpretation of data. Maybe the last DNA generate profile it become very improper. Their interpretation, their meaning will different. Become the faulty laboratory procedure will happen also, right? Some machines to exhibit that process DNA fingerprinting that will have some defect. Then their procedure also became very wrong, right? That is only. Now satellite DNA and microsatellite, what is that? Satellite DNA is consist of long stress. Remember, consist of long stress of repetitive DNA sequence alongside in the tandem. 5 to 4% there in humans, and factors. Key feature of the cell DNA that is highly repetitive. One is highly repetitive. Next, AT means adenine and thymine rich sequence. Those sequence nucleotide based pair. Those are highly is there and mostly non-coding. They are become non-coding to any other sources and structural role is also there. The classification and type of satellite DNA here. Satellite DNA's classification alpha, beta, mini satellite, microsatellite, centromere. Alpha satellites was we observed near to the centromere. I will show where it. Let me. This is assume this is chromosome. This is chromosome and this is centromere. Okay, and beta satellite a pericentromeric region. This is pericentromic region. Okay, this is pericentromic region and this is what is that centromere, right? And mini satellite is telomeres. Those round circle I denoted, right? These round cycles, those are telomeres. There will we can observe this kind of mini satellite telomeres and microsatellite throughout the chromosome. And it is 1 to 10 base pair and base pairs will vary and high base pair is there in alpha particle that is centromere at the present of centromere. You know that. And microsatellite DNA. Here micro means small. Just remember micro means small. Small here microsatellite are short DNA sequence. Short. Just remember short DNA sequence. Here satellite. Here satellite it means long stretch of repetitive DNA sequence is there. But here short DNA sequence. Here short tandem repeat will be the simple sequence and characteristics there. It's possible to high mutation rate and highly polymorphic and usually non-coding and useful genetic markers. Here application it is help for DNA fingerprinting. Unique repeat pattern is there, so we can use DNA fingerprinting and genetic diversity. The population and evolutionary biology. We can know the genetic diversity between the peoples and this is research. We can got to know that because of this, that is repetitive, small and short, right? Is there another sequence of DNA? Because of the sequence of DNA, we can detect the genetic disorders and which part of the genes will performing that disease, contribute to the disease. Here, now DNA barcoding and eDNA. Here, guys, DNA barcoding is a technique used to identify species using a short standardized DNA sequence. Here, how the barcode will represent, here some analogies there. Like it's one example that is supermarket barcode identifies product, right? Some barcode will be there on the product, right? Product item they will scan it and then they will know to the value of that product. Like that, DNA barcode identifies the biological species. Standard marker gene used in barcoding memorize. So for animal, centro the cytochrome C oxidase first is the mitochondrial DNA. It will present in the mitochondrial DNA. And plants, MART-K and RBCL is there, two standard plant genes. Chloroplast. In plants, DNA is there in chloroplast. Okay, the fungi, ITS region, internal transcribed spencer. Okay, nucleotide ribosomal DNA is there. Type of barcoding DNA sample is there and tissue sample, bulk sample, and environmental DNA. Okay, DNA extracted directly from single organism tissues. DNA extracted directly from one single organism tissue. Here, bulk sample means multiple organisms we need to for aquatic insect collection. For example, here environment DNA obtained directly from water, soil, or air. Okay, environment DNA means water, soil, air. Advantage: no need to capture or harm organism here. Application of the bird DNA coding is species identification and biodiversity assessment, wildlife conservation, detection of invasive species, analysis of animal diet, identification of pollinator, and medical plant ID and food authentication. Authentication. I hope you can enjoy this. And these are some terminology there. You can read it for for better revision. Okay, let's we go. Biotechnology, biology plus technology, right? And modern biotech here, genetic engineering, DNA editing will be there. Human genome means 20,000 to 25,000 genes there in human DNA, right? DNA present in nucleus and mitochondria for animals. Sir Alex Jeffrey, 1984. He founded the fingerprinting piano. Okay, PCR polymerase. The polymerase. Sorry, but wait, I forgot its name. Okay, where we are stopped. The polymer chain reaction. That is DNA amplification too. Okay, polymer chain reaction will help to spread of the DNA in it too. Okay, and microsatellite. It is short tandem repeat and plant barcoding MART-K RBCL are present in the chloroplast. And eDNA it is direct identification without capture. Here, traditional biotechnology, the known data on a DNA manipulation is their gene, functional unit of hereditary, right? And human DNA length is 3.3 into 10 power 9 base pairs there, and adenine thymine is 2 hydrogen bonds, and guanine and cytosine is 3 hydrogen bond is there. Identical twins. Identical twins DNA fingerprinting and those repeated gene sequence is there that will be similar in the twins. Okay, and satellite DNA 5 to 10% of the human genome, and animal barcoding CIC gene is there, and fungi barcoding ITS region is there. Some questions is there. If anybody you want to answer here. Okay. Hmm. Here it will be answer will be showing. If you want, everybody answer it. Okay. And don't try to crime all the data what are there in notes, and please be very authentic in your preparation, and I hope you are preparing very good. Let's end up the sessions, and I will welcome in the next session of I will taking next session also biotechnology also. I will show that is 100 standard means advanced biotechnology there. Hello guys, I said you in the last class I will again come with biotechnology. This is second section of biotechnology. Okay, not section. This is our second session about biotechnology, and here we are going to learn about chromosomes, RNA, transcription, translation, microRNA and RNA. Okay, let's go to the what is chromosomes. I hope guys, before that I want to tell you one message. Please try until you get it. Okay, never be afraid to ask anything for anyone. We are not able to ask anyone, then answer will be always no. So first try to ask anything, whatever it is wrong, right, anything. But try to ask. If you ask, then only you get to know the answer or something different that will help you to achieve something somewhere in your life. Okay, let's go. Let's go to our chapters. Okay, that is biotechnology part 2. Next, we are going to learn about chromosomes. We in the last section we learned about DNA fingerprinting, DNA barcoding, right? All those we learned and their application also learned. Today we are going to learn chromosomes. What are the definition? It is a thread-like structure. Just assume it is a thread-like structure. I've shown you, right? This is the chromosomes. Okay, this chromosome. Okay, this is a thread-like structure, and it that chromosome will present inside the nucleus. Okay, inside the nucleus. That will present inside the nucleus. Wait, I got here some marking point. And nucleus cell that carries hereditary information in the form of DNA. That chromosome contain DNA, right? DNA has the capacity that carry the hereditary, right? That will also carry hereditary information in the form of DNA. It is packaged form of DNA it has, okay? And structure of chromosomes, DNA is extremely long, right? DNA is so many long, okay? Means and to fit inside nucleus, just remember, to fit inside nucleus we need to pack very tightly now. For that, the tightly coiled by a histone protein, that the coiling tightly packed, okay, tightly packed of the DNA in chromosome because of the protein histone. Just remember histone, because of the histone, the DNA in the chromosome, they are tightly packed. And some, okay, just remember, cell is there, consider take a one cell from your body, just assume, okay, and after that, in the cell we have nucleus, right? In that nucleus we have chromosomes, in that chromosomes we have DNA, in that DNA we have genes. That's it, this is the diagram, okay, hierarchy diagram. The component of chromosomes, components, what are the components? Just we have two components here, one DNA and the another one is histone protein. That histone protein will help to packaging the DNA in the structure, okay, in the chromosome, and structural support, it will help to structural support, and any gene regulation also it will help. What are the functions of chromosome? It will carry genetic information and transfer hereditary traits also, right? Traits means some characters. Okay, height, weight, all those. What are the important character of our body and all those information? And ensure accurate cell division. It will also help to accurate cell division, right? And control protein synthesis through genes. It will control protein synthesis through gene. For example, we have in somatic cell, right? Our human somatic cell contains 46 chromosomes. There are 23 pairs, right? In that 23 pairs, we have 22 pairs are autonomous and one pair is the sex chromosomes. Okay. And now we are going to learn about we learned about chromosomes, right? Now we are going to learn about RNA, that is ribonucleic acid. Definition of ribonucleic acid. It is nucleic acid. Here in the name only itself is there. Nucleic acid is there. To try to remember here. Okay, nucleic acid. Nucleic acid involved in gene expression. Okay, means it will the RNA will help to the formation of the protein. So, and it has nucleic acid in RNA. We have nucleic acid in RNA, right? That will help in gene expression and protein synthesis. Generally, it is single stranded. Just remember, it is single stranded, not like DNA. And each nucleotide, each nucleotide has, the nucleotide will bind here because of the ribose sugar and the phosphate group and nitrogenous base. Here, nitrogenous base has adenine, guanine, cytosine, uracil. Here, purines are adenine and guanine. Pyrimidines are cytosine and uracils. Here, RNA contains uracils. Okay, instead of thymine. Thymine is present in the DNA. Okay, this difference you have to know. Okay, the structure and feature of the RNA here. Structure and feature. Okay, here features of what is the their sugar is the ribose sugar is there in RNA strand. It is a single stranded base. Uracil is there instead of thymine. Stability is less stable than the DNA because DNA is a double stranded. It is a single stranded. Here length is shorter than the DNA. What are the functions of RNA? Just remember, guys, functions here, and it is very important, okay, to know everything. So it will help to the protein synthesis, transfer genetic information from DNA to ribosomes, and genetic material. Second one is genetic material. In some viruses, for example, HIV, influenza, and coronavirus, TMV, tobacco mosaic virus. In that also we have see, remember, coronavirus, tobacco mosaic virus, influenza, HIV. These virus has their genetic, our genetic, the human being genetic material is DNA. Okay, viruses has RNA as the genetic material. Enzymatic activity here. What is enzymatic activity? Do you know RNA will act as a catalyst or enzyme? Not a catalyst, it acts as a enzyme. Okay, means that will enhance the reaction will be further faster. For example, ribozymes, that is that enzyme is called ribozyme. Here type of RNA is also there. Those are messenger RNA is there and ribosomal RNA is there. Transfer RNA is there. Okay, messenger RNA, it will help to the carry genetic instruction. It will carry genetic instruction. Just remember, it will carry genetic instruction from from where? DNA. DNA to ribosomes. It will carry genetic instruction from DNA to ribosomes. Okay, that's why it is messenger carrying instruction. Ribosomal. Second one is ribosomal RNA, right? Now form ribosomes along with proteins. It will form ribosomes along with protein. It responsible for the protein synthesis. With this in the cell in and let me know tell you the before both the conclusion, and I want to share what is the transfer RNA here also. Transfer amino acid to ribosomes during protein synthesis transport vehicle. Okay, transfer amino acids to ribosomes during protein synthesis. It will help to the transfer amino acid to ribosomes. See guys, here ribosome. The ribosomal will help to the protein synthesis. In the protein synthesis, we need amino acids. Okay, to amino acid for the protein synthesis also we need. That amino acid will transfer in that ribosomes, that will through the transfer RNA. So these ribosomal RNA, transfer RNA will help to the protein synthesis. Okay, just remember like that. Now we are going to learn about the central dogma of molecular biology. Okay, here just in a brief, just just in a brief, you understand, from the DNA to it will DNA is converted as a RNA. RNA it will convert the protein. Okay, RNA will synthesize the protein. Okay, DNA will form as the RNA through the help of transcription. From the RNA to protein synthesis will help the translation. What is transcription here? We are going to learn about now transcription, the process by which genetic information stored in DNA, right? Here, genetic information stored in DNA is copied into the messenger RNA. Okay, just remember, some genetic information is there in DNA, right? That information will that information will carry by messenger RNA. Okay, that will, and that's located in the nucleus. That is enzyme involved here, RNA polymerase. In the transcription, enzyme, that is RNA polymerase enzyme will help. Okay, just remember which enzyme will RNA polymerase. Steps of the transcription here. Step 1, DNA unwinds. Step 2, RNA polymerase binds DNA. Step 3, complementary RNA nucleotide added here. And messenger RNA is synthesized. Then messenger RNA exits nucleus. Significance here. Protect DNA, remain in nucleus, and allow protein synthesis as a temporary copy. First step in gene expression. Do you know, right? In our nucleus, there is a double stranded DNA, right? When we need to synthesize the protein, right? It will, when it will act like single, okay? It will go further like that. So here, breakage of the nucleotide base will be happen between that breakage, and this will form single stranded became a, that is RNA, okay? This will transcription, and with the in the process of transcription, that is RNA polymerase enzyme will help, okay? And those there also the messenger RNA will be synthesized, okay? That will exist in nucleus only. This process is happening in nucleus only, okay? Allow next here, DNA protects, okay, remain in the nucleus only. Allow protein synthesis as a temporary copy, first step in the gene expression. Now transcription. No transcription, it is transcriptome. What is transcriptome? You all are, if any are science background, if you all this studied in the our 12th standard, okay? Complete set of all mRNA molecule produced in a cell or tissue or organism at a given time. You know, right? What is that? Complete set of all mRNA means we have here complete set of all mRNA in the cells. Cells not, that is transcription happening the process. Then mRNA will be synthesized to carry the message from DNA to RNA, right? So there will be synthesized that complete set of mRNA produced in a cell or tissue organism in a given time. That is transcriptome with the help of transcriptome. It is genome, here transcriptome. What is the difference here? It is complete in genome, complete genome DNA is there. The transcriptome, complete mRNA is there. Just remember transcriptome. Last M and M here. Try to remember, okay? Remembering structure I am saying here. Just for remember, transcriptome mRNA is there. Genome, full complete DNA. Genome means whole DNA sequence, right? Just remember like that. And it is stable and it is dynamic. We don't know it is dynamic, okay? Maybe sometime, some after time it will become a stable. So it will vary time to time. Same in almost every cell, but it changes with cells type also. Their structure, mRNA structure may be changed with the cells to cell. Here permanent, the genome is permanent, here temporary, right? Till the mRNA will present till temporary. Why? Because exist till it will exist till the formation of the protein. After that, it will again, when it will form protein, their work is done. They will go wherever they want. Don't like that. After that work is this finished, they will not active. And importance here tell us which genes are active, cell functions and development stage and response to environment. Here important. It those these means these transcription, right? Transcriptome it will help to which genes are there are active and what are the cells functions and development stages also, and response, how they are responding to environment also. Here translation. Okay, it is very interesting, guys. Keep waiting. Not keep waiting here. Just be active. Okay, it is very interesting topic. Definition here. Covering genetic information in mRNA into the protein. See, we did the stages till mRNA, right? We did still from the mRNA. Now we are going to learn how the from mRNA to the protein synthesis that will help to the process of transcription. Okay. Ribosomes into cytoplasm. Just remember, in ribosomes, ribosome is placed in the cytoplasm. Okay, a molecule involved in mRNA also, transfer RNA also, ribosomal RNA also, and amino acid. Amino acid will carry with the help of the transfer RNA, right? RNA, ribosomal RNA is there to protein synthesis, and ribosome is there in the cytoplasm. Here, messenger RNA we did in transcriptome, right? In transcriptome, mRNA is there. Step here, mRNA reaches ribosome. See guys, mRNA is reaches ribosome, then transfer RNA bring amino acid, and also amino acids joined together form protein form like that only. It's very simple. Okay, protein performs structural, enzyme, hormonal, and immune functions also. Now, microRNA. What is that microRNA? Have you heard? If you know anybody microRNA, before going to I am explaining, just do in the comment section. Just what you know, just add the in the commentation in your own words. Okay, don't to try to crime here. Don't to look here. Okay, it will help you very well. And also, if anybody are not interesting to write any notes from here, just try to write the what I am saying in comment section while listening, or after listening one paragraph, pause that video and try to analyze, and also try to write it in comment section, because it not help for me, because it will help to remember you for a long time. Okay, let's go. I am deviating, right? Why I am deviating? Okay. Now microRNA. Here, small non-coding RNA that regulate gene expression after transcription. Here, micro non-coding RNA that regulate gene expression. Okay, that will regulate gene expression after transcription and control protein production without coding for proteins. Okay, it will help to the formation of the proteins without coding of the protein. Okay, it will control the protein production. That's it. There is no further here. Okay, here's some Nobel Prizes got. Okay, just try to learn out. In 2024, Nobel Prize in Physiology and Medicine is there, awarded to Victor Ambers and Gary Ruvkun. Why? Why they got Nobel Prize? Because discovery of microRNA, its role in post-transcriptional gene regulation. Okay, after post-transcriptional gene regulation, they have they discovered the microRNA because they will control the protein production without coding for any protein. Okay. Sorry. Okay, let's go to learn why important it is. Although every cell has identical DNA, right? All cell has identical DNA. For example, brain, muscles, liver cells, they perform different function due to gene regulation. How microRNA works? Let's assume guys here DNA is there, and after the DNA, mRNA will form, right? Synthesized in transcriptome, right? MicroRNA binds mRNA. Just remember, microRNA will bind mRNA, that will protein production blocked. MicroRNA if bind with mRNA, then protein production will be stopped. Result, translation stops. Okay, translation will be stopped. Protein not formed, gene expression reduced. Protein is not formed, gene expression is reduced. Let me know clarification here. I said right here. Okay, wait. From DNA to RNA, that is transcription, and RNA to protein, that is translation. Okay, just remember. And now listen here. The translation stops means from mRNA to protein process, that will stop when microRNA binds to the mRNA. Okay, the protein synthesis will be become less and gene expression will also reduce. That's it. The characteristics, they have very small. It is non-coding and highly conserved, found in plant and animal. Okay, it is very small. Okay, it is non-coding, highly conserved. If it is coded, why it is non-coding? If it is coded, then protein will be not formed, because of it is non-coding. Okay, because of that it is non-coding and it is highly conserved, found in plants, and animals, and discovered using model organisms, Canaro habits elegans. C. elegans and in roundworm. Importance biomarker, the control development and cell differentiation. Okay. Development also cell differentiation it will control because it is binding to the it will uh, not enable to the formation of in a body. Just try to assume in our body protein is not formed. The protein is very important nutrient for us, right? To grow. It is not able to develop that protein because of the binding of microRNA. Then it will control our development and cell differentiation rate also. A cell will not differentiate again right here and seen in because micro see just we observed in normal human we can't observe microRNA, but we can see in the cancers and diabetes, cardiovascular, neurological disease, viral infection. So excellent biomarker for the cancer diagnosis. MicroRNA is very excellent for cancer diagnosis. Here, RNA interference. Wow, now it is interesting character, right? RNA interference. What is that? Okay, biological process. It is a biological process. Just remember biological process means any bio within some process is happening, any reaction like that. Where RNA molecule, just remember RNA molecule suppress gene expression. You know, right? Like that microRNA that will reduce the right that will reduce protein formation. Like that, it will also suppress gene expression by destroying or blocking mRNA. Okay, here it will destroy that mRNA and that will the messenger RNA will carry the messages from the DNA to our RNA in the ribosome that will black or destroy that mRNA only. That is the interference and the post-transcriptional gene silencing. It is also known as the post. Yeah, after that, right. It is the first DNA to RNA that process is what transcription, right? That's why it is post. After the transcription only it is happening, right? Post-transcriptional gene silencing or gene silencing. RNA interference is the gene silencing and post-transcriptional gene silencing. Principle here: destroy mRNA. No translation will be happen, no protein, no so gene silent will become chup like that. Okay, gene silent will become. So protein also not formed. And translation also is not happening here. mRNA also not there. Why it is? Interference RNA has like this much capacity. It will not help to the protein formation in our body. Major molecule. Okay. Small interfering RNA and second microRNA. Application of interference RNA is antiviral defense. It will become antiviral defense means protect against virus and foreign genetic material. Okay, it will be active when right when any virus will become in our body it will protect from any genetic material. Okay, foreign genetic material if came in our body it will protect us. Yeah, this is good, right? Now it is playing very important role, right? Because virus came in our body and that will lead to the proliferation of their genetic genes. Sorry, their genetic material will proliferate in our body. That will control by this interference, RNA interference, right? So it is very helpful as okay. Gene regulation, yeah, it control development and differentiation. Yeah, this because of this only we are protecting us, right? And scientific research, understand gene function. It will help to the understand gene function. Medicines, it will help for therapy for cancer, viral disease, and genetic disorder also. Agriculture, development of pest resistance and disease resistance crop. In agriculture, it will help the pest resistance, also disease resistance crop also. Okay, some differentiation there, microRNA to small interference RNA. The source is it is naturally produced. It is natural or artificial. Okay, interference RNA it is present in our body also. Natural is also there, but artificial it is prepared in artificially. Okay. Function here regulate gene expression here gene silencing. It regulate means it will control means yeah regulate means control only, right? It will also control gene expression. It regulate gene expression, but it completely destroy. Okay, that is complementary. It is partial and it is usually complete. It will complete process and target is many genes specific gene. It will target for specific gene only. Means those when viruses enter in foreign body enter in our body, then only it will become a reactive. Micro versus RNA. It is small and it is small RNA molecule biological process. Regulate gene, silence gene, same disease. It is natural regulator and this is uses microRNA and sRNA and block translation and degrade mRNA. And I hope guys you enjoyed this session also. And okay, this is some revision session is there. Okay, let's go to try to do this also because it helped too. Okay, I will tell you the word and if you want to before I'm saying you can write in a comment section. That's why it will help you to remember in a long time. Okay, chromosomes. What you remember? Just try to write in a comment section. Chromosomes. What is the chromosomes? It is present in the DNA, right? In DNA, in nucleus, it is there, and in DNA nucleus, in nucleus, that chromosome is there, right? That is that chromosome had DNA package because through that DNA package has that is histone protein. Just like that you write there. RNA has RNA has ribose sugar and also uracil and it is single stranded. Messenger RNA, right? It will message and RNA, ribosomal RNA, T is transport amino acid. Transcription, yeah. If I said transcription, what is happening? DNA is converted into the RNA. That means RNA, right? That is mRNA. That is inside in the nucleus only. It is happening inside in the nucleus. Okay. Transcription happening inside in the nucleus. Translation is happening outside the nucleus. Means happening at the ribosomes. Ribosomes where is there? Here only translation is happening. Means mRNA into the protein formation is right. Transcriptome. All mRNA molecule in a cell at a given time. All mRNA molecule at a given in a given at a time. Transcriptome means all mRNA will be the right whose the messenger carrier mRNA molecule at a given time. All means all mRNA in a given time is there. That is transcriptome. 2024 ಅಲ್ಲಿ Nobel Prize they got the Victor Ambers and Gary Ruvicon for because of the micro identity discovery of the microRNA. MicroRNA binds mRNA and that will stop prevent the protein synthesis. RNA interference. It is a post-transcriptional gene silencing, right? And it is key molecule. MicroRNA also small interference RNA. RNA is useful in the first resistance crop, antiviral defense, and medicines. I hope guys you enjoy this session. If you need for this like this very small notes okay compared to other any coachings and this will help to clarify if you not understand with my word also if you read this you can clearly understand okay and there is no wrong here okay and I hope you are not writing anywhere just write in a comment section because just because for not for me I'm saying because it is helping you because I also did so many things for like that only for comment section for comment to comment or not for comment to again I am saying current comment okay for current affair also I am doing same I if I listen anything I will try to comment there itself what I gain from that do that exercise because it will help your muscles, okay, muscles memory, and that will help to develop your answer writing also. And I hope guys you are enjoying, and please be very active in learning. Don't be very disappointed and don't be read in a partial manner, okay? Be very active. Okay, bye bye, I will see you in the next session.